Hi, so in this video we are gonna talk about the spleen, and as we know the spleen is one of the secondary lymphoid organs as we said in the previous videos. So the spleen consists of two parts, the white pulp and the red pulp. This is the white pulp, significant handwriting, and this is the red pulp. Okay, let's talk about the white pulp first. Here is the artery, comes from the splenic artery, then divides into arteriole, and it's surrounded by this structure which is called peri-arteriolar lymphatic sheath, PALS, and this contains the T lymphocytes. After that we have the follicles, and if you remember from the previous video of the lymph nodes, the follicles are primary and secondary. Primary if the B cells are active and they are dark, and secondary if the B cells are active and it's pale because of the germinal center. Okay, and so these are the components of the white pulp: the follicles, the peri-arteriolar lymphatic sheath, which is surrounding the arteriole coming from the splenic artery. Here we have a marginal zone between the white pulp and the red pulp, and its name is also marginal zone. So easy. Okay, what does it contain, or what's its function? It contains the macrophages, and macrophages, as we know, functions as a filter for the blood components, for any debris or dead cells. Also, it can act like antigen presenting cells for the T cells, T lymphocytes in the PALS. Let's talk about the red pulp. The red pulp, from its name, its color is red, and it's rich in blood supply, and it's very hemorrhagic, and that's why any injury to the spleen is very dangerous, and it can be fatal from the severe hemorrhage which can occur. So it consists of two things: the splenic sinusoids, these splenic sinusoids are open. This means that they let to all the blood elements to pass through them to be filtered in this structure which is called the splenic cords. What are the components of the splenic cords? It contains macrophages for the filtration of RBCs, of platelets, of any blood component, any free antigen, and so on. Let's draw some macrophages like this. Okay, and I think that's fair enough for the spleen. Let's talk about a condition called splenectomy. A splenectomy can be done for many reasons. The first one is traumatic. After an accident, as I have said, it can cause fatal hemorrhage, so we need to remove the spleen. A second reason is the ITP, immunothrombocytopenic purpura. Many patients improve after removing the spleen, because the spleen is the site of destruction of platelets, as we said in the red pulp. Also, a condition like spherocytosis and sickle cell disease. In sickle cell disease, it's functional as splenia because of the sickle cell crisis. We will talk about this condition later, which causes dead parts for the spleen until it's completely non-functional. So we consider it as splenectomy. So what happens after the spleen is not present anymore? The first thing, the patient will be susceptible for infections with encapsulated organism. Why encapsulated organism? Because the encapsulated organism resists destruction by phagocytosis, so they need special deal. What is this deal? Here we have a macrophage. Let's draw some teeth. Evil, right? Also, so we need first a macrophage, and we need antibodies binding to this organism. These antibodies will cause two things. First thing is activation of complement system, which is a group of proteins, system of proteins, causing damage to the encapsulated organism, and also it can cause opsonization. What's the meaning of opsonization? It's a process for making phagocytosis easy to the macrophages, a component of the complement system called C3b. Also, opsonization can happen by the binding of the antibodies. So here we have the antibodies and the complement system causing death to the encapsulated organism, and also the complement system causing opsonization for the organism to be phagocytosed by the macrophages. So if we don't have a spleen, first of all, we don't have macrophages, which are present in marginal zone and the red pulp. Second thing, there will be a lack of antibodies because of the lack of B cells in these follicles. We will lack the complement system, because complement system activation depends mainly on the antibodies. Also, we will lack one component of the complement system, C3b, which is important for the opsonization. All of these reasons will be a cause for the susceptibility to encapsulated organism infection. What are these encapsulated organisms? First of all, we have strep pneumonia, and also we have the meningococcus and the Haemophilus influenza. So a patient who is having splenectomy should receive vaccination to these encapsulated organisms. Something called Howell Jolly bodies. I hope I spell it in a right way. What are these bodies? Normally, some RBCs exit the bone marrow having nuclear remnants, a small percentage which is filtered in the red bulb of the spleen. That's normally occurs, but if the patient is lacking the spleen after splenectomy, then we will see all of these RBCs, nuclear remnants, abundant in the blood smear, and it will seem like this. That's what we would see in the blood smear. Another thing is called target cells. These are RBCs with something like that, a crescent shape. So what are these target cells? They are RBCs with large surface area of what are these target cells? They are RBCs with large surface area of cell membrane. They are normally produced from the bone marrow, but also normally they are filtered in the spleen through the red pulp. In this case, after splenectomy, we will see them abundant in the blood smear. Another thing we would see, thrombocytosis. And I think it's easy. We would see many platelets in the CBC, or complete blood count. As we have said before, the platelets are filtered in the spleen through the red pulp. So after splenectomy, the patient would have high number of platelets, which is called thrombocytosis. That's all about the spleen. I hope you understood it. If you have any question, leave it in the comments below and I will answer you. Don't forget to like, subscribe, and turn on the notification button for any new video. See you in the next video, and goodbye.